Hello, good day. I'm Joseph Aisan Jr., currently affiliated with the Southeast Asian Fisheries Development Center, Aquaculture Department in the Philippines. And on behalf of my co-authors, I'm going to present on disease and pests in eucheumatoid seaweeds, which is the two major limiting factors affecting the sustainability of the seaweed aquaculture industry in the Philippines. Seaweeds have been an important aquaculture commodity in the Philippines. The Philippines started the commercial production of eucheumatoids in the late 1960s or early 1970s, and through the years contributed significantly to the economy, either as a source of dollar revenue or a source of livelihood to thousands of families engaged in seaweed farming. In 2018, seaweeds comprised 64% or equivalent to 1.48 million metric tons of the total aquaculture production in the country, making seaweeds the top aquaculture commodity by volume and second by value, supporting an export industry for Carginan worth 207 million US dollars. The seaweed production in the Philippines reached its peak in 2011 when the country produced 1.84 million metric tons. However, in the following years, a decline in production was observed. In 2011-2012 production volume, a 4.9 decline in production was observed, worth 32 million US dollars. Among other reasons, the following are the factors that contributed to the decline in seaweed production in the country. First is natural disasters such as typhoons that were observed to be stronger and more frequent in occurrence in the last decade, resulting in widespread destruction in the farming areas throughout the country. Second is the limited supply or availability of quality plantets or seedlings throughout the major seed farming areas in the country. The third is the reported outbreaks of disease and pests. The two commercially important disease and pest problems observed in the farms are the epiphytic filamentous algae or EFA and ice disease or IID. Both IID and EFA outbreaks have been reported to affect biomass production and the quality of carrageen extracted from affected seaweed plants. EFA and IID have affected the seaweed industry of the country. However, data relating to temporal and spatial distribution, seasonality, prevalence, and etiology of these critical problems at the farm level are still lacking. This study aims to document the signs of pests and diseases in several major seaweed farming areas around the Philippines and quantify the occurrence of IID and pests, including AFA and macroepiphytes. Second, to evaluate the severity of IID and EFA at selected farming areas and report the significant potential for yield limitation due to these conditions within country. For the methods, seaweed farms to be assessed were chosen based on the biomass output in the area, accessibility and availability of assistance and a local person as a guide during assessment activities. The following information was gathered from the farm owners or operators: the culture technique, species farmed, the number of cultivating lines per farm. The farm assessed include Palawan in Luzon with 5 farms, Bohol in the Visayas with 5 farms, and Zamboanga City, Davao del Norte, and Tawi-Tawi in Mindanao with 6 farms. The total number of farms assessed was 16. Based on this information, 4 to 5 cultivating lines to which individual seaweeds were attached were randomly selected for assessment, representing at least 5% of the total individual seaweeds at each farm site. In situ visual inspection for the presence of IID and pests was made on plants from each line. Assessments were conducted during low tides for fixed off-bottom farms, and a motorized boat was used to access farms in deeper waters. The prevalence of pests and IID was calculated. Severity cover was computed on 3 farms that exhibited the highest IID and EFA prevalence. For each sample, the apical, secondary branch, and the primary branch of an individual seaweed thallus were assessed. The presence of IID in AFA was estimated as percentage cover using a semi-quantitative scale from 1 to 5. The total score per region of tissue was divided by the number of seaweeds assessed to obtain a per plant severity score, and classified into 4 infestation covers: low, moderate, high, and very high. Water parameters were analyzed in situ and samples for nutrient analysis were collected. Additional climatological data was also collected from the nearest synoptic station. For the results, environmental parameters varied between each farm. Seaweed were exposed during low tide in shallow water farms, while for farms at deep water sites the depth ranged from 2.5 to 8 meters during assessment. Regions range from 120 to 2,670 micromoles photons, with salinity in the range from 27 to 34 psu. Temperature ranged from 27.3 to 32.2 degrees centigrade. pH from 7.6 to 9.1, and dissolved oxygen from 5.3 to 9.2 ppm. The variation in readings of the different parameters will be influenced by the time of day and weather conditions at the time of sampling. For example, reagents reading in Bohol were high because of dense heat and clear skies during the sampling period. For the nutrient analysis, at the farms, phosphate levels range from not detected to 0.015 ppm, nitrite 0.04 to 0.07 ppm, nitrate 0.10 to 0.38 ppm, and ammonia not detected to 0.38 ppm. The highest volume of rainfall during the sampling month was recorded in Visayas farms, with 223.6 mm, and the lowest monthly rainfall was observed in Samal, Davao del Norte, with 60.8 mm. The mean monthly wind speed ranged from 2.3 m per second, and near surface air temperature ranged from 28.4 to 29, with the highest recorded at Samal Farm. Seaweed farms were found either in shallow or deep water areas. Both the shallow and deep water farms were represented in each region. 4 culture techniques were employed: triangular, 1 farm; fixed off bottom with 7 farms; hanging long lines with 4 farms; and multifold rough long line with 4 farms. The number of seaweeds varied from each farm and ranged from 49 to 3,000 seaweeds per line, depending on the length of the cultured line and the culture techniques employed. Cultivation density, or the number of seaweeds per meter of line, was 4 seaweeds per meter for all farm sites studied, with the exception of 1 farm in Palawan, where the cultivation density was 10 seaweeds per meter. Caphophycus alvarezi, Caphophycus striatus, and Eucheuma denticulatum were the common seaweed species being cultured. Disease and pest observations were described depending on its type association to the host seaweed. Type 1, if there is an observed changes in the thallus tissue. Type 2, attaches loosely to the surface of the host but no damage is observed. Type 3, if there is an observed penetration to the seaweed intercellularly, injuring the cortical and the medullary cells. And type 4, observed damage or absence of tips on the tissues of the thallus. Type 1 association were observed on ice disease and black spots affected seaweeds. IID is described as loss of pigmentation followed by the softening of the thallus and detachment of diseased plants from the seaweed cultivation lines, while black spots are black or brown raised spots 0.5 mm in diameter on the surface of the eucheumatoid thallus. Type 2 association with the whole seaweed includes macroepiphyte algae and described as seaweeds that grow on the surface of the seaweed plants, either as loosely attached or entangled. Examples of these macroalgae are brown alga sargassum, green alga ulva, and red alga gracilaria. While the type 3 association includes epiphytic filamentous algae and described as red seaweeds that grow on the surface of the seaweed plants, penetrating from the cortical to the medullary layers, causing mechanical damage to the host plant. And examples of these are Melanotamnus and Polysiphonia. The last type association is the type 4, which include grazing and observed as mechanical damage or absence of tips on the soft tissues at the apex of the thallus. For the ice disease, in Mindanao, the Sama farm recorded a very high IID prevalence of 100%, but overall, the prevalence was classified as low. For EFA were observed in 4 of the 16 farms assessed, including Quezon, Guindacpan, Tictabon, and Arena Blanco. All the farms exhibited a low prevalence of EFA. For macroepiphytes, the presence of sargassum, ulva, and gracilaria was observed in Luzon and Mindanao farms. However, no macroepiphytes were recorded on seaweeds observed at the Visayas sites. Sargassum was present only in Luzon at 4 of the 5 farms. Filamentous ulva species were observed in Luzon and Mindanao farms at 3 out of 5 and 2 out of 6 farms respectively. Gracilaria was observed only in 1 farm in Mindanao with a low prevalence rate. For the black spots, they were only observed on plants collected from 1 farm in the Visayas with a low prevalence. Grazing. Signs of grazing were recorded in 2 shallow water farms, one in the Visayas and the other one in Mindanao, though the prevalence at farms in both areas was low. Also, no significant difference in IID prevalence was observed when shallow water at 5.0% and deeper water at 14.8% farm sites were compared. The relatively high prevalence of IID observed in farms located in deeper water than in shallow water farms, albeit not a significant result, was unexpected as the shallow water farms are more exposed to fluctuations in environmental conditions. Many studies of IID to date report on the occurrence of the condition in shallow water farms, and there is a lack of information on the occurrence and progression of the condition in deeper water farms, and no information available on the occurrence of IID outside of the cultivation settings. It may be that prolonged high or low rainfall volume, low wind speed, and high near surface temperature in deep water cultivation sites contribute to a stress response in seaweeds, resulting in a development of disease signs. Changing weather patterns brought about by climate change may also influence seaweed disease occurrence in deep water farms. The results of this study may provide new insights that IID is no longer confined in shallow water farming areas, but also occurs in deep water farms when unfavorable environmental conditions continue to persist over an extended period of time. A very high severity cover of 76 to 100% was found affecting the secondary and primary branches of affected plants. IID was observed in different parts of the thallus, suggesting that any part of the seaweed is susceptible to IID. These results may imply that in environmentally stressed seaweeds are more susceptible to colonization by pathogenic bacteria. However, no data exists relating to the full diversity of microbial taxa associated with eucheumatoids in either healthy or diseased states, and the process behind the development of IID signs remain unknown. The EFA severity cover was observed to be low, affecting the whole thallus of the plant. When observed microscopically, EFA penetrated up to the cortical cells and created injury to the host plant, thereby providing an entry point for pathogenic microbes, resulting in secondary infection. EFA outbreaks in seaweed farms may result in biomass loss and compromise carcinoid quality. For the summary, this is the first study to compare the occurrence of important eucheumatoid disease and pests across major seaweed farming areas in the Philippines. Ice disease occurred in all regions surveyed and across all cultivation methods and cultivar samples, while macroepiphytes and epiphytic filamentous algae were not reported at all sites and when present were typically at moderate to low levels. However, as each site was sampled only once, and therefore it remains to be seen how the prevalence of pests and disease changes over growth cycles and seasons, and how this responds to fluctuation in environmental conditions, including salinity, water temperature, and nutrient availability. It should be a priority for future research to address this lack of knowledge. Further studies should also aim to increase our understanding of the relationship between eucheumatoids and their associated microbial communities, the structure and the function of which may play a role in the development of disease signs. A greater understanding of the factors influencing disease and pest occurrence will inform proactive management strategies, which are becoming increasingly important as disease and pest outbreaks become more frequent. The result of our study can be found in our recently published paper with the title Preliminary Survey of Pests and Diseases of Eucheumatoid Seaweed Farms in the Philippines. This study was conducted under the Global Seaweed Store Project, a consortium of research and academic institutions in the UK, Tanzania, Malaysia, and the Philippines, funded by the United Kingdom Research and Innovation Global Challenges Research Fund. Thank you very much for watching and have a nice day.